In order to begin talking about fossils, we first need to discuss the basic dating methods that are used in order to determine how old they are, so that as we begin talking about fossil primates, you have an idea of how they came up with these dates. Most of our evidence on fossils comes from the field of paleontology, which is the study of fossilized animal and plant remains. Now, fossils form whenever organic material gradually dissolves and is replaced with minerals that crystallize out of the groundwater. It's a relatively rare process, but when it happens, we can observe the remains of things that lived millions of years ago. Now, within anthropology, we have the field of paleoanthropology. So it's sort of combining paleontology with anthropology. In order to practice paleoanthropology, most paleoanthropologists will employ the following approach. They work on dating the site, determining the paleoecology or the ancient environment of that site. They look for archaeological traces of behavior, that would be material remains like tools. And then they look for any anatomical evidence from hominid remains, so trying to find bones from hominids that may have lived there. The entire study sort of relies, again, on the idea that fossils are usually mineralized remains, but also the study of taphonomy, which is how bones and materials can become fossilized. A very small number of bones fossilize. I mean, less than 1% of what's on the planet will ever fossilize. So we really have to look closely at how that process works in order to understand what we're seeing. Here's an example of the way a fossil could form. So for example, let's say 1.05 million years ago, we have a dead hominid here. At about 1 million years ago, we have an eruption that sheds volcanic ash over the dirt in which the fragmented skeleton of the hominid is buried. Over time, that surface erodes and we end up finding the fossil buried between two different layers of volcanic ash. This would allow us to determine the date of the layer that those bones were found in, because we can very easily date volcanic ash even if we couldn't date the bones themselves. Now, anytime a paleoanthropologist starts working, they look for context. So what's the environmental setting where we found things? Can we recreate that paleo environment, which we'll talk about more in the next lecture? In order to date samples, there are sort of two different ways to go, and it just depends on the type of sample and the context that you find it in, which one you can do. We have relative dating techniques, which will allow us to determine whether an object is older and younger than another object that we can date easily. And we also have absolute or chronometric dating methods that will provide us with an estimate of age in years based on radioactive decay of certain elements. So let's look at some of each of these. In terms of relative dating, we have stratigraphy, biostratigraphy, paleomagnetism, and fluorine analysis. Stratigraphy is based on the idea that you can study the way that deposits of earth layer in a sequential fashion. And it's based on the law of superposition, that a lower layer should be older than a higher layer, which should make sense if you think about it. The higher layers are the ones that are more recently deposited. So if we look here at the layer where X is versus the layer where Y is, based on the law of superposition, X should be the older layer. So if we can date something in Y, and we find something in X, we know that it's older than what we found in Y. So again, that's where that relative dating comes in. A lot of times if you look at sedimentary rock, you can clearly see the layers because they change as their mineral content changes. So this helps with relative dating. Now, biostratigraphy is similar, but it's more of a faunal correlation. So correlating what you find with the other animals that you find in that sequence. So for example, if we know that animal A is 1,000 years old and we find something right next to it, that thing is probably 1,000 years old as well. Paleomagnetism is a dating method that's based on the shifting of the geomagnetic poles. So you know how we have a north pole and a south pole? Well, those are actually magnetic poles, and they shift every so often. Positive becomes negative, so on and so forth. What happens is they actually cause the ions in rocks that have iron in them to shift direction. So if we were to look at volcanic rocks, you can see, depending on where the poles are at, the rock themselves actually take on a different appearance. So if we find things in a layer of rock that looks like this, below all of these other layers, it tells us how old something is. Fluorine analysis allows us to analyze groundwater For example, the longer a bone has been deposited, the more fluorine it's going to have in it. But we can only use this analysis to compare it with bones from the same location. So for example, to figure out if two sets of bones date back to the same time in the same place. You can't compare fluorine analysis from one site to another. Now, chronometric or absolute dating, as we said, is more exact, but we can only date things that have radioactive decay of unstable isotopes in them. What that means is that over time, an isotope like carbon-13 or carbon-12 or carbon-14, an isotope will disintegrate and form another variation of another element. That allows us to then look at the pattern that we see. The rate of disintegration for the different isotopes is different for different elements, but follows a distinctive pattern. So basically, carbon decays at a different rate than potassium or argon. So it allows us to figure out, based on the sample we're looking at, how much disintegration has occurred, how long did it take for that disintegration to occur. Depending on the half-life of the isotope that we're looking at, so the time that it takes for it to decay, we can date things anywhere from billions of years ago, like the age of the Earth, to less than 1,000 years. Some of the examples of this would be potassium-argon dating, uranium dating, carbon dating. We'll look at two of these a little more closely. Carbon-14 dating is used for organic samples like bone, wood, charcoal, shell, and peat. Carbon has a half-life of about 5,700 years, so we can date from approximately 100 years back to 40,000 years, and sometimes even back to 75,000 years. But if we want to look at something that's much older, we would use potassium-argon or argon-argon dating. These are going to be in volcanic rocks, so volcanic ash, certain igneous rocks like basalt. Because potassium-argon elements have a half-life of about 1.25 billion years, we can use them to look for anything between 1 and 5 million years ago, although we can find earlier dates in other parts of the world. So these are just two examples of how dating with an unstable isotope can work. Now, molecular dating is used looking at the DNA of the organism that we're interested in. So we can try and develop a molecular clock. We can compare the difference in the DNA between two samples and use a calculated constant mutation rate to figure out when they diverged from each other in history. Now, there's obviously problems with this because mutation doesn't always occur at a constant rate, but it can still give us a good ballpark. We can also use DNA hybridization. What that means is we basically heat up the DNA from two different species and allow it to stick together. So you have one strand from one species and one strand from another species. When they heat up, they separate, and when you cool them down, they'll stick together with each other. And depending on how well they're attached to each other, we can figure out how similarly they are related to each other. Basically, the more strong attachment they have, the higher the temperature it takes to break them apart, the more closely they must be related. So that's another thing that sort of allows us to look at genetic similarity, which can be helpful in molecular dating.